All right, ladies and gentlemen, welcome back. We're going to be looking at part one of DNA. So this is all about structure of the DNA, and also we're going to get into the process of DNA replication. All right, so let's get a pen up here, move on. So we're going to be looking at the structure first of all. So this is just a little cartoon on the interpretation of the structure, the 1953 when it was discovered. You will have to give more detail than this, so you can stop it and read this one. Okay, so let's look at it. We talked about this before in nucleic acids, that DNA is made up of nucleotides. So when we have a string of nucleotides together, that's what's making our DNA. Little ladder here. This is a terrible picture, right? So let's look at it. What does it consist of? So first of all, it's got this phosphate sugar backbone. We call it phosphate and deoxyribose. That's where the deoxyribonucleic acid comes from. More specifically, it's a phosphoric acid, and more specifically, it's a pentose sugar, which means 5 carbon chain, carbon ring actually. And the last one is the nitrogen bases. And we know that the G's and the C's go together, and the A's and the T's, so I'll just skip ahead to this. So G's and C's go together, and A's and T's because they're complementary. But we have this new term, and what it means is a purine versus a pyrimidine. There's just the types, they're named based on their structure. So both the A and G have this double ring nitrogen based structure, whereas the pyrimidines just have a single ring. And you'll see this in the next picture coming up. So if you look here, so this here is what we call a purine. And this one is a pyrimidine, and it's just based on how many rings, carbon rings they have in their nitrogen base. Okay, so just look at the structure of DNA. So we have our alternating phosphate, sugar, and we call this part the backbone, and there's 2 sides. And then what we have is the nitrogen bases. So here's adenine mixing with a T. We have, what is this, cytosine mixing with guanine. We have thymine and adenine, and we have G and C. So they're going to be mixed together. What actually joins them together is a hydrogen bond. That's what joins those nitrogen bases together. All right, so DNA structure continued. We talked about that phosphate sugar backbone. In here, it's a little bit more simplified. We have our deoxyribose sugar here and our phosphate. So again, just makes up the sides of the ladders. 2 strands twisted into a double helix. This little shape is called a helix. So when we have 2 of them, it's called a double helix. And like I said, the thing that creates this force, this attraction between the bases, is a hydrogen bond like we talked about. So we have this polar end of one side and the polar end of the other is going to want to join. So H on one side and the OH of the other, or I guess just really the O. And as we know, if we look at DNA, certain sections of that DNA is going to make up a gene. And what do genes do? Gene is what's going to actually determine the end product of a protein. So the big thing here, what we're looking at with protein synthesis, DNA, is the making of a protein. The way that the DNA is arranged is going to tell us specifically what protein is going to be made, and that relates to all the different functions in the body. All right, so now that we know the structure, sometimes we're going to have to replicate this DNA. So we're going to have to make the exact same stuff. So why would we want to do that? So first with some background, replication just means turning 1 DNA helix into 2 DNA helices. The reason why we'd want to do this, if we ever get hurt and have to repair our cells, in order to get a new cell, you want to have the same DNA in it, so it needs to replicate. Also, if we want to grow, in order to have any growth, cell division, we need to have identical cells. And like I said, each cell needs an extra copy, so that's why we need 2 sets of DNA. One thing might be new from the last time you've heard about DNA is that there's a conservative, so ignore most of this for the most part. Semi-conservative means that here's our original DNA, and what's going to happen is it breaks apart. Okay, so each strand that's going to form, so here's your new strand that's forming, right? You're going to have 2 new strands from that first one, but each one of the new strands has an old piece that we use to as a template. Okay, let me just erase this. Okay, so that's the semi-conservative part. Okay, it just means that each new piece of DNA will have an old piece that was used as the template. Okay, let's look at the process now. This looks a little bit complicated, and it is, but there's pretty much 3 enzymes that we care about. The first one is DNA helicase. As you can see here, it's this triangle shape here. It's a protein, specifically an enzyme. What it's going to do, it's going to catalyze this reaction to break apart those DNA bases. So it's really unwinding and unzipping the helix. The next one is DNA polymerase, which is right here. What it's going to do is it's going to take these DNA nucleotides, which are floating around, I'll just put nuke, these DNA nucleotides, and it's going to grab them and it's going to start pairing them up one by one in a complementary fashion to that existing template. Okay, so it's going to do it by the DNA polymerase. And then the third one is called DNA ligase, and all that's going to do is it's going to fix any errors or missing sections in the DNA, and we'll talk about why that's going to be the case in a second. Okay, so you're going to want to know the 3. So we have helicase, DNA polymerase, and ligase. Okay, and we'll talk about some ways to remember those. So just an overall, and just one extra thing as well. So as you can see, it shows the arrow. We have these 5 and 3s. All it is a reference point in terms of structure of those DNA molecules, but it's also direction. DNA is going to always be copied going 5 to 3. But since they're complementary, the other side is going to be 3 to 5. So this is going to be copied this way. So the problem is that when you open this part up, it's going to expose these new nucleotides. So these can be deposited one after another this way. But as you can see, when you're opening it up and you have to start going with the direction, every time you have to backflow, get back again every time it opens up in order to produce those new nucleotides. So what happens is sometimes there's gaps. And what we do to fill those gaps is we use things called Okazaki fragments, which are fragmented DNA that actually get stuck in there. You don't have to worry about here. All I want you to know is that the Okazaki fragments, OK frags, replace DNA gaps. Okay, so you don't have to worry about the RNA primer or primase. That's something that you'll get into in university. Okay, so hopefully that's okay. We'll be doing a lot to work on it because there are some tough concepts. What do you need to know? 3 parts of nucleotide, 4 types of complementary bases, and then the 3 enzymes, which we have HDL, helicase, DNA polymerase, and ligase. Okay, have an excellent night.